I would like to invite Professor Imanol Amayra and Mercé Pallares on stage to hear about cognitive profiles in the CTNNB1 profile. They come from the Deusto University and they've been working with Spanish children for a while, and they bring a lot of interesting information for everyone. Thank you. Oh, thank you. Estivaliz, Ana. I am accompanied by Mercy and the rest of our team who are sitting back there, including Francisco Lopez and Maitan. This was a first research paper we created as professors working with rare diseases, neuromuscular rare diseases. And we only learned about the specific features of CTNNB1 during a symposium, and we had the opportunity to visit families with children with CTNNB1. And based on that experience, we gathered a set of cases through different methodologies. We spent 3 or 4 days with them. Maybe Merce stayed longer. I'm sure she did. And after that, we gathered data, established a protocol to determine whether there's any specific feature to all children with CTNNB1 versus other children with neurodevelopment disorders. So I know that we're running late in the agenda. Our sample is tiny, merely for descriptive purposes, but we have studied this sample thoroughly. We took many hours to go into the data, and we thank the parents who are here for providing this information. Usually, a psychologist cannot spend full days studying this kind of material, and we're very grateful for it. To understand this paper, we need to understand that there was no prior information on this disorder, and we can only base our study in better known animal disorders. And one of the revelations of this paper is precisely that perhaps a possible conclusion is that biological research is more important than clinical research when it comes to determining the cognitive profile for these children. Experiments on animals conclude that in the case of CTNNB1, alterations experimentally produced in lab mice and also upsets in the signaling path for a glycoprotein, which is WNT with beta-carotene, which is the synthesis of the fundamental component of CTNNB1. In other words, a child with CTNNB1 basically has an upset in this glycoprotein. And if the levels of this protein are altered, the child, or in this case, the lab mice, exhibit difficulties to exhibit fear responses. In other words, they are fearless mice and therefore can display aggressive behavior repeatedly because they do not learn that in the face of harm, they need to display a defensive behavior. Also, when these lab mice, and we will relate this to children later, but when these mice are given a substance inhibiting the possibility for this protein to be affected, the mice do not recognize the space around them. In other words, they cannot create a visual space map of their environment. And that's a very specific feature that affects a very specific part of the brain. It's a hippocampus, which is related to memory. We know that we have a left and a right hippocampus. The left one allows us to remember words, and the right one allows us to know whether we need to move forwards or back. So if these children and these mice have a hippocampus impairment, they get lost and they don't know how to get back, and they exhibit repetitive behavior. And then further studies, not on mice, but on humans, that have no CTNNB1 but some impairment in the CTNNB1 gene shows that these subjects have a slightly lower IQ than expected in terms of neurodevelopment. However, subjects with CTNNB1 gene alteration but not CTNNB1 disease do not necessarily have behavioral issues, which is the case in this syndrome. Also, unhealthy patients, by altering this, provisionally altering this gene with a protein, it has been observed that healthy patients, anyone in here, if anyone should have that gene altered, you would lose your episodic memory. The things we need to say, the things we need to think, the things we said a few minutes ago, the things we need to do some days from now, that capacity to remember is lost. Therefore, episode memory in healthy subjects is temporarily affected by altering CTNNB1. But also, when other abnormalities are present in this gene, we see Alzheimer's, schizophrenia, or Huntington's. Altering this gene generates memory dysfunction. So, animal and human models studied so far do seem to indicate that an alteration of the proteins coded by this gene exhibit behavioral and memory disabilities. And then going to the cognitive phenotype, out of all the material we went through and the bibliography we had access to, there seems to be a big clinical problem that I would like to present here. True evidence, tests, or methodologies that can be repeatable and would lead us to understand the intellectual level of children in CTNNB1 are restricted to this slide, whereas there are other papers reporting that there's an IQ of around 27, but none of those studies describe how these numbers were obtained. So, why are so few researchers telling how intelligence has been measured in children with this disorder? We understand that these children were not assessed under scientific methodology, and these data are merely empirical. Somebody concluded at some point that these children had an average IQ of 50. We believe this is not only inaccurate, but also poor practice. So the cognitive phenotype profile at present exhibits these issues or these shortcomings. There are no appropriate measurement devices. Second, no common methodology is applied in most research papers, and therefore they're not repeatable. There's no general IQ to show the phenotype variability. There's no breakdown on the type of intelligence. And finally, there are no previous studies about the difficulty of conducting cognitive evaluations of these children. Only 13% of them could actually go through the test. Thank you, Imanol. Next, I will explain how we did our study. Based on all the previous considerations, we also considered the following. We went through the Simon-Simplex collection. It's an international protocol for neuropsychological and psychological tests specific for ADHD children. We also reviewed the bibliography on disorders related to CTNNB1. And last, we considered that it was important to pick cognitive tests that were short and specific, assessing specific features in a simple, specific way. So the purpose of our paper was identifying cognitive and behavioral features in CTNNB1 based on the following tests. The Child Behavior Checklist. It allows us to identify adaptation and behavioral problems SCQ, or Social Communication Questionnaire, detecting possible autistic features, and then the vocabulary Peabody test for spatial relations and receptive vocabulary, and a clinical study that would allow us to gather behavioral features in CTNNB1. So the following step was contacting with the Spanish chapter, the CTNNB1 Association. Parents signed an informed consent form, and the questionnaire was addressed to these parents. Based on the WebForms platform, we applied the two questionnaires and also a clinical study, and during approximately 1 year, we've been meeting with parents to provide face-to-face tests and assessments. On screen, you can see the data variables with a mean deviation of 3.2, average age 7. So we drafted the main clinical symptoms of the disease, and we ranked them in the following graph. First of all, about neonatal development. The first few symptoms or alterations detected by parents include not meeting development milestones in 80% of cases. Also, during neonatal development, there's significant presence of foot disorders or eating disorders and signs of microcephalia. In terms of motor development, there is significant retardation in acquiring motor abilities, as you have heard in several presentations, and also movement abnormalities in 100% of cases. As for sensory levels, there's visual dysfunction. 90% of cases report vision problems, out of which many required glasses. Also, these children have excessive reaction to sound and noise and little tolerance for pain. Also, sleeping disorders are pervasive, and many of these children are already medicated to deal with the symptoms, so only 50% of cases actually reported sleeping problems. And out of that 50%, the highlights come from persisting sleeping problems and difficulty to fall asleep, and then problems in activation and awareness. As for eating disorders, there are several types of those. The clearest one is the fact that these children ingest non-edible objects, and also for sphincter control, there's late control milestones. And then about language disorders and speech disorders, there's a problem in the acquisition and development of natural language, particularly the capability to articulate. And last, in emotional terms, 100% of cases show that these children are easily irritable. And then there's a clinical assessment, which is vital. One of the main symptoms detected out of the features in this disease is related to cognition and behavior. It is therefore important to analyze the symptoms. Here's a list of the score and the test. We start with a Peabody test described based on 11i. As you can see, 4 out of 10 cases get an IQ above 70, which is considered the borderline score at which you can start about intellectual disability. However, these tests are based on a non-clinical population, and those scores you see on screen, which is the minimum score, are actually affected by the bias of a lack of a sound description of neurodevelopment disorders. Now that as these children understand better than the tests indicate. However, we don't have the same results for visual space relations in the Raven test. As you can see, in many cases, percentiles move away from the mean or the average, which exhibits difficulty to understand and to perform the tasks required by the Raven test. Then SCQ indicates the possibility of autistic features. After or above a score of 15, we can claim that there are autistic symptoms. And last, on CBCL, the most usual behavioral issues in the syndrome are externalizing in nature. And to conclude, our paper attempts to provide a new methodology breaking down intelligence into two different units, which hadn't been reported before. We only heard about general IQ, and we didn't even know what it meant. We don't know if this was based on abstract numerical or verbal reasoning. And then our IQ, it turns out, our actual IQ found is better than expected, particularly based on understanding language. They understand better than it seems, or better than others seem to think. Also, there's an expression that may negatively affect performance in tests. Therefore, we need to be careful. You need to be careful when you talk to professionals and tell them that these tests cannot be applied on these children as they are applied on healthy children. Obviously, the children are forced to speak and articulate in these tests, and obviously these children cannot deliver that information. So since there's bias on this variable, understanding is better than expected. It greatly exceeds the scores in other studies or papers, and the Peabody test had not been applied. And also we see the same results as in animal research. And therefore, animal research seems to be the best target method to determine visual space relations in these children in the Raven test, as happens with animal subjects. And the question now is whether this is merely related to the right hippocampus or whether it can be associated to an exudative retinopathy problem that was pending for specialist evaluation.
So our results do not normally coincide. They don't normally coincide because intelligence was higher than expected, and the Peabody, which is the test that we used, should be a test of choice because it doesn't require the child to say anything. The child just has to answer certain stimuli, and so this is a way of understanding listening comprehension. And so listening comprehension is something positive that these children have. Of course, they do have autistic symptoms, most of them, but not as much as we expected. And of course, we need more extensive examinations. And I would insist that we are inspiring our work not on what psychologists do, but on what neurobiologists do, especially in Italy. In Italy, there is a team of neuropsychologists that have really done very, very important things that will be applied to this CTNNB1 syndrome in the future. Thank you, Imanol, Mercé.